I believe this is going to be part 7. We'd gone through kind of an intro, we'd gone through all the steps in animal-human virus replication. The last video we were talking about how viruses generate genetic diversity and why that's significant to humans. So this next one, folks, we'll just very briefly talk about why it's so challenging trying to treat viral infections, why we don't have as the diversity of drugs as we do, say, with bacteria. And then we'll go into different categories of viral infections. Okay, so this is slide 29. So we'll start here. And one of the questions is, why is it so much harder to treat viral infections, say, compared to bacterial infections? So one of the challenges is we want to remember that the viruses are using our cellular biosynthetic machinery to replicate themselves. And so if you want to stop viral replication, you would worry that you would also be shutting down biosynthesis within your own cell, and that's going to harm your cells. So this is kind of a challenge, trying to find true magic bullets for viruses, magic bullets being antiviral drugs that would inhibit or stop the replication of the viruses without harming your cell. So again, this is kind of the point here, right? That a lot of the antiviral drugs do actually have pretty severe side effects, okay? And then another concept is that we want to remember the viruses are replicating within our cells. And that means that the viruses are living in a little bit of a protected fort. The cell membrane, any cell membrane is going to act as a diffusion barrier against what are usually hydrophilic antiviral drugs. So you might have high concentration of antiviral drugs outside of the host cell, but maybe there's lower concentration of antiviral drugs within the host cell. So the fact that the viruses are replicating inside our cells also makes it a little bit more challenging. These are just, this is just one example of a family of antiviral drugs. The nucleotide analogs, also called nucleoside analogs or base analogs. So these are antiviral drugs they're look-alike. So these are the normal nucleosides that would be used, say, in replicating DNA, right? So what the chemists have done is they take the normal nucleoside, in this case, you guys, so remember this is a nucleoside, and then they chemically modify it. And usually what happens is then when the virus is replicating and it incorporates that nucleoside analog, that look-alike, it will either stop the viral nucleic acid synthesis, just stops it, or the introduced nucleoside look-alike will cause really, really, really high mutation rate, right? So that most of the viruses most of the newly replicated viruses would be no longer infectious, right? So, but again, you guys, the problem is with a lot of these, like our cells, like our DNA polymerases might use these nucleoside lookalikes, right? So that potentially could cause harm to our cells, right? So just a couple here, folks. I remember when HIV was first discovered and everyone was just so worried about it. The first anti-HIV drug was this azithromycin, or AZT, which is right up here, AZT. So that was the first anti-HIV drug. And then acyclovir is a common drug right up here. Acyclovir is a common drug used to treat, say, herpes virus infections. And then what's not shown up here are a couple of drug classes that are used against HIV. So we have HIV protease inhibitors, we have HIV integrase inhibitors, we have, with HIV, we have co-receptor antagonists that will help block the attachment of HIV to our cells. So this is just one class of drugs here. right? And there's other classes of drugs that we can use to treat the viruses. AZT, you guys, just because I think it's cool, AZT, its target is the HIV reverse transcriptase, right? So that's going to hit that unique HIV enzyme, the HIV reverse transcriptase. Oh, and just on that note, you guys, I know there's been, again, going back to the pandemic coronavirus, people have been talking about chloroquine maybe being used to treat the pandemic coronavirus. And so I got out the pharmacology book. So chloroquine's been used since the 1940s to treat Plasmodium, to treat malaria. And I was just interested in why might chloroquine help inhibit an RNA virus. And it's a mystery to me, you guys, because it says here in the pharmacology book, the mechanism of action for chloroquine remains controversial, so they're not sure why it works in Plasmodium and malaria. And it says, and obviously you guys, you don't have to know this for the exam, but chloroquine probably acts by concentrating in parasite food vacuoles, preventing the polymerization of the hemoglobin breakdown product heme into hemozoin. And the buildup of the heme is apparently toxic for the Plasmodium. And so that's one model for how chloroquine can kill Plasmodium, kill malaria. It's classified as a 4-amino quinoline. And so it doesn't, to me, you guys, it doesn't look like it's a nucleotide or nucleoside analog. So it's, if it, I just don't know why chloroquine would help stop the replication of the pandemic coronavirus. So obviously that's going to take a lot, much more research, many more studies. Okay, folks, so we're going to keep going. So we're going to leave treatment for right now. And now, folks, we're going to talk about different types of viral infections, like what is the result when the virus invades. So this is really dramatic, of course, you guys. So some viruses are what we call lytic or cytocidal viruses, and this would be really similar to the T bacteriophages that we talk about. They can only carry out the lytic cycle. So cytocidal, cyto means cell, cidal means to kill. So these are viruses that are going to kill our cells. And one way they can do it is through shutting off the biosynthesis that our cells require for, to maintain themselves, to repair themselves. And so the viruses can come in and take over our cellular biosynthesis. So all of the biosynthetic products are only going to be for the virus. So viral nucleic acid, viral proteins, right? And then our cells can't take care of themselves and they end up dying. And then another reason for these cytocidal animal viruses, if they cause a lytic infection, right? After replicating, they cause our cells to die, to break open. And you guys, of course, I was trying to get a really dramatic one. This looks almost like a vampire. Makes you wonder if maybe some of the vampire stories come from poor people that were infected with, say, adenovirus. So this adenovirus, it can cause cytocidal damage, right? And in this case, the sclera of the eye, it's just bloodshot red from damage to all the capillaries. So maybe that's where the vampire stories came from. So folks, now this is different, a different type of infection. So some viruses, when they invade us, we might have an initial bout of clinical signs and symptoms. So we know we're sick, right? But then what those viruses can do is following that the clinical signs and symptoms, we might feel like we're over it, right? Thank heavens, my immune system took care of that virus. But what we might not understand is we are still infected with the virus. So these infections we would refer to as latent viral infections, or we talk about latency. So latency, one way to describe it would be like a dormant infection, right? The viral genetic information is still within our cells, but the viruses aren't replicating. So during that time, we won't have any clinical signs or symptoms, right? But the bad news is there can be some signal that triggers reactivation of the virus. So now it is going to start replicating again. And that will usually trigger another round of clinical signs and symptoms, right? So again, we might think that we're cured, we got rid of the virus, and then it could be even years later, the virus will reactivate, right? And we'll have another round of clinical signs and symptoms. So some examples of viruses that can cause latent infections, and these would be, you guys, good lecture exam 3 questions. So the herpes virus family, it's a huge family of viruses. So some people are shocked to know that the chickenpox virus is a herpes virus. So as kids, we might get infected with chickenpox, right? And then we recover, but we're latently infected. And then later, when we get older or stressed, or especially when we get old and our immune system starts to fail us, the chickenpox virus will reactivate. And the clinical signs and symptoms are described as shingles. So the fancy name, you guys, for chickenpox shingles, varicella zoster. So I think of varicella. Um, Varicella is being the initial infection causing chickenpox. Zoster is the reactivation of the chickenpox infection causing the shingles. And I think officially, you guys, you call the chickenpox virus, you call it the varicella zoster virus, right? Um, so that is a tough one. And then another of the herpes viruses, you guys, when I was growing up, or I shouldn't say growing up, but maybe when I first started studying microbiology, we learned these as the human simplex viruses, type 1 and type 2. Now officially they're called the human herpes viruses, type 1 and type 2, but often I forget and I call them the herpes simplex viruses, type 1 and type 2. These are brilliant at causing lifelong latent infections. Oh, and you guys, I have to share this with you. So several years ago, a medical microbiologist, a professor emeritus of medical microbiology from the UC Davis School of Medicine, he came to give our infectious diseases course a talk on sexually transmitted um, pathogens. And he was very distinguished, came in suit and tie. He had a lovely British accent. He had um, um, been to medical school, I believe, in Shanghai, and very serious, right? And so he starts the lecture by asking, "Now, class, why are herpes viruses like diamonds?" And I was like, "Oh my gosh, I have no idea what the answer is." And then he said, with a perfectly straight face, he goes, "Because diamonds are forever." And again, folks, there's no disrespect, because these herpes viruses, once we get infected, they cause these lifelong infections and can cause a lot of suffering. But sometimes people will use kind of this dark humor, right, when we're talking about tough subjects. But do remember that, you guys, herpes, a lot of them are like diamonds because diamonds are forever. We'll come back and talk more about the human herpes virus type 1 and type 2, and we'll come back and talk more about the varicella zoster virus. Another group of viruses, you guys, that we need to remember can cause latent infections are the papilloma viruses. And you might think of papilloma like wart, so these are the so-called wart viruses. Different animals can have different papillomavirus infections. So we'll focus on the human papillomavirus, the HPV. And we want to remember, you guys, there's lots of different strains. There's strains that can cause just common warts, say on your hands or the bottom of your feet. There's strains that can cause genital warts. And then we'll talk about some of these strains, you guys. They cause latent infections, and they can actually increase our risk for certain types of cancers. So some of these latent HPVs are what we call oncogenic, right? They can cause cervical cancer, cancer of the penis, anal rectal cancer, and now we're starting to see cancers of the throat where these HPVs are causing latent infections. And then, folks, a third group of viruses that can cause latent infections are the retroviruses, and the most notorious one, of course, is HIV. So when HIV, it once it gets into our cells, you'll remember it carries out reverse transcription. It uses its RNA genome as a template, and in a two-step process using HIV reverse transcriptase, a double-stranded DNA copy of HIV genetic information is made. We call that the HIV provirus. That HIV provirus inserts itself into our chromosome, and once it gets in there, we can never get it out again. And it might be that for some time, maybe the HIV provirus, it's not directing HIV replication, but then sometime in the future, that cell might get activated, and then the HIV provirus will reactivate and trigger HIV replication. Okay, so again, folks, herpes viruses, papillomaviruses, retroviruses can all cause latent infections. So folks, what we're going to do here is we're going to use a couple of herpes viruses just to maybe give some more details. So we want to remember following the initial infection, and sometimes with the initial infection we'll have clinical signs and symptoms. Maybe sometimes they're so mild we didn't even know we got infected, right? But following that initial infection, then the viruses, at least their genetic information is going to hang out inside of our host cell. And again, they aren't replicating, so during that period, we won't have any clinical signs and symptoms. Depending on the virus, the viral nucleic acid may or may not insert itself into the host chromosome. And do remember, you guys, when we were talking about Henrietta Lacks and her cervical cancer cells, it was discovered that it was an HPV strain 18 had inserted its viral DNA into the chromosome of her cervical cells, and that is what had triggered them to become cancerous. So that can certainly be a problem if the virus inserts its genetic information into our chromosomes, because that might increase the risk for cancers later on. So these latent viruses, right, they're just kind of resting, hanging out. We want to remember that months or years later, they can be reactivated. We don't understand all the triggers for reactivation, but with reactivation, the viruses now start replicating again. And that's going to cause more clinical signs and symptoms. And you think, gosh, I thought I was immune to that virus, right? Or you just didn't realize you'd remain infected. And again, you can remain infected for life. And this is just kind of maybe kind of nerdy, you guys, because there's some viruses like hepatitis B virus that cause persistent infections, meaning once they start replicating in our liver, they're constantly replicating, constantly replicated. Then we would contrast that to a reactivation of a latent infection, where there's periods where the viruses are not replicating themselves, and then they reactivate and start replicating. So you guys don't worry about this. I wouldn't ask this on the lecture exam. So let's look specifically at the human herpes viruses type 1 and 2, also known as the herpes simplex viruses. So these two types, folks, historically, type 1 would cause infections above the belt, right? So we think of fingers, especially if you go into dentistry. We think of the mouth, the face, the eyes, right? So usually those are type 1 herpes viruses. And usually, but not always, the type 2 herpes viruses are going to cause the genital tract infections, right? But you guys remember, herpes viruses do not read our books. So type 1 herpes viruses can cause infections above the belt, right? So infections of the head, infections of the finger, and type 1 herpes viruses can cause infections below the belt in the genital tract, right? And this might be of importance when you're talking to your patients that, say, for example, have maybe a type 1 herpes infection, say of the mouth, that maybe just need to educate your patients that they need to be careful when they're being intimate with their partners, right? Because these viruses, they don't really care where they're growing. Okay, so folks, again, the first infection is the primary infection, and very often with the herpes viruses, often you will get lesions, and the herpes viruses cause a lytic replication. So what will happen is you'll get these fluid-filled vesicles that are very painful. I have a type 1 infection in my mouth. They're really very painful, and what makes it worse is that the fluid inside those vesicles is chock full of infectious virus. Okay. And a sneaky way that the viruses can move from one cell to another, sometimes they can trigger our cells to fuse together, and that lets them move from one cell to the other without being exposed to the immune system. So now with the human herpes virus type 1 and type 2, after they replicate in the epithelial cells, right, say of our mouth or of our fingers or of our genital tract, then they invade the sensory neurons. Isn't this amazing, you guys? They invade the sensory neurons and they travel up the axon through the cytoplasm to where the neuron cell body is, where the nucleus would be located, and they just hang out there, right? And again, this could be for lifetime. So for me, you guys, I in college I got an oral type 1 infection. I was so sick. My whole mouth was like one big blister. It took me a long time to get over it, and I was like, well, thankfully I'm over it. But what I now know is the viruses had invaded my sensory neurons and had traveled up to the trigeminal ganglion, right? So the trigeminal ganglion. And now that herpes virus is permanently with me, hanging out in my trigeminal ganglion. And what I've noticed is times where I'm under lots of stress for long periods of time, or I've also noticed like if I get too much sunlight, like on my lips, too much UV, my virus will reactivate, and what it does, and it's so weird, you guys, I can almost feel it. It moves back down the axon, and I experience this as kind of a tickling, and this is so weird, you guys. I think of the virus, it's like marching down my axon, and then it reinvades epithelial cells that those sensory neurons are innervating. The virus reinvades epithelial cells and starts another round of lytic replication. And again, you guys, so painful. The bad news is I'm probably shedding viruses in my saliva even before I'm aware it's reactivating. So that's something of concern, right? Now, for folks that are going to go into dentistry, dental hygiene, dentists, what if you're a dental hygienist or a dentist and you're working on me in my mouth, right? And I'm shedding herpes viruses, right? I'm not aware of it. There's no lesions yet, but I'm shedding herpes virus. And let's say with one of the dental instruments, by accident, the dental hygienist or dentist, they nick their finger, right? They have gloves on, but those dental instruments are sharp, and they break the skin, they break through the glove, they break through the skin, and now herpes virus from my saliva has gotten into their fingers, right? So this is really tough, man. So this is a occupational hazard of dentists and dental hygienists. The fingers epithelial cells are going to get infected with what's called herpetic whitlow, right? So that would be the primary infection. And then the viruses, they invade the sensory neurons, they travel up here, and then they're going to hang out for the rest of our lives in the brachial ganglia. And again, you guys, when there's a trigger, they'll reactivate, they'll travel back down and reinvade the epithelial cells, causing again these horribly painful lesions. And what makes it bad then, you guys, is then the dentist or dental hygienist can be shedding herpes viruses, right? So that would be bad for their patients. Okay. And then, folks, if we are first infected in the genital region, here the same thing. We'll have these really horrible, painful, fluid-filled lesions, and then eventually the immune system responds. But the viruses have invaded sensory neurons, and they're going to go hang out up here in the sacral ganglia, right? And again, with reactivation, they're going to travel back down, reinvade the epithelial cells, another painful reactivation of the herpes virus. And again, folks, this is really important that we educate ourselves and our patients. Again, if somebody has a latent herpes infection, and we have reactivation, the herpes virus can be shed even a couple of days before the patient is aware that they're having reactivation, before the patient experiences any clinical signs or symptoms. And that's why it's important that folks that have so-called genital herpes, that they're really, really conscientious about safe sex, right? Protecting their partners, because you don't want the partner who you love and with whom you're intimate, you don't want them to get infected if you can help it, right? Um, one thing, folks, just because this came up, if 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 a person has, say, genital herpes, or maybe really, really harsh, severe herpes on the face, and they're having lots of reactivations, and we'll talk about, you guys, we're really worried about ocular herpes, right? Because repeated reactivations can actually cause blindness. There are anti-herpes drugs, and one of them, I think one of the most famous one, is acyclovir. So one of my students, this was many, many years ago, she shared with me that she had genital herpes and that the doctor kept her on acyclovir. Now, it won't cure us, but it will really decrease the viral replication. So that means it should hopefully reduce the reactivation. It should reduce maybe the number of painful lesions, maybe hopefully reduce the pain of those lesions. And very importantly, you guys, it will help reduce the number of viruses that we're shedding, right? And again, especially if we're being intimate and we have genital herpes, we want to be really careful that we're protecting our partners, right, with whom we're being intimate. So there are anti-herpes drugs, but remember, it won't cure us, but it can keep viral replication greatly reduced. These are just some examples, folks, of the human herpes virus type 1 and type 2. This is kind of the classic, folks. So this might be an initial infection, or this could be the reactivation. This is what I get all the time. Usually it happens right before the semester begins, so I get to greet my new students with these wonderful reactivation of my herpes virus infections. Sometimes these are called cold sores, and I'm not sure if it's because people associate them with the onset of some other infection like a cold, right? But again, folks, very, very painful, fluid-filled vesicles, chock full of viruses. So you just want to be really, really careful, because do remember, you are infectious at that point. So for example, you wouldn't want to go to the dentist at that time. Be really careful, you guys, kissing newborn babies, because newborn babies can actually get infected, right? If we're kissing them around their head, their face, so we want to be really careful there. This is of huge concern, you guys, because we can see that this herpes virus has infected the eye, right? And then once those, I think there, it help me out, you guys, the ophthalmic branch, um, sensory neuron gets infected, then this person could have reactivation, repeated infections of the eye, and that can cause blindness. Here is the herpetic whitlow, the occupational hazard of dentists and dental hygienists, right? Very, very painful. This is called herpes gladiatorum, after the Roman gladiators, who were these, fierce warriors that would go into the Colosseum and battle to the death. So herpes gladiatorum, this is kind of an occupational hazard for folks involved in contact sports like wrestling, MMA, where people are in close contact with an opponent, and there's like kind of trauma going on to the skin here, right? So this poor guy probably at some time had an opponent that was shedding the herpes virus. Maybe it was like just from the mouth, right? And so he has infections here on the skin of the face, up here by the ear, almost into the scalp, and very, very tough, you guys. He's got a herpes ocular infection. So I would think with herpes ocular infections, I don't know, you guys, I'm not an expert, but I'm wondering if they might treat you maybe more frequently with acyclovir, because you really don't want multiple reactivations, right? And then, folks, this would be an example of herpes infections of the genital tract on the penis. And again, folks, these vesicles chock full of infectious viruses, very, very painful. Oh my gosh, so painful. And again, we just want to make sure, if there's one partner that it has a herpes genital tract infection, right, we just want to be really, really careful about safe sex and protecting our partners so that they don't get infected. And again, folks, the bad news is with genital herpes, often you're shedding before you have your first signs, your first clinical signs or symptoms. So we just have to be really, really careful. But again, you guys, all of these, in theory, this and that especially, can be treated with acyclovir to reduce viral replication. Of great concern is if a pregnant mom has a genital tract infections with herpes viruses. And the big concern is if mom has just very recently acquired the genital herpes infection, if she's pregnant. And the reason that's of huge concern is that if mom's pregnant and she very recently has been infected with herpes virus, she won't have time to make protective antibodies, maternal antibodies that will cross the placenta into her baby, right, to protect the baby from an overwhelming herpes infection if the baby is delivered vaginally. So my understanding is, folks, that our big concern if pregnant mom genital tract infection with herpes, if she's shedding at the time the baby is delivered, and if the baby is going to be delivered vaginally, because it's going to be in the, for example, the vaginal epithelial cells are releasing the herpes virus. The baby, as it comes through the vaginal canal, is going to be bathed in herpes viruses, right? And because the baby, their immune system isn't fully developed yet, the baby's immune response is so minimal that it will be really difficult for the baby to control the herpes infection, and the herpes can spread everywhere. You know, it can invade the skin, the eyes, spread throughout the whole body, and even invade the central nervous system. So with neonatal newborn herpes, depending on the study, we see mortality rates from 30 to 80%. So 30% mortality is 30 babies out of 100 would die. Out of every 100 babies infected with herpes, 30 would die. And then this is horrible, you guys. So 80% mortality is of every 80 babies infected with herpes, 80 of them would die. And you might, makes you wonder why such a range in mortality rate. Well, it might have to do with when mom got infected. Because let's say mom has been infected for several years with genital herpes, she's had a chance to make protective antibodies, neutralizing antibodies, right? And there's one class of human antibodies, folks, that can cross the placenta, transplacental transfer, and that's the best antibody of all, IgG. So if mom's been infected for a long time and she's pregnant, she'll transfer those protective IgG antibody across the placenta into the baby. So when the baby's born, the baby has mom's antibody circulating and helping to protect against herpes infection. So that might be why we see a big range here, right? Another thing, you guys, I have to remind myself, because I do have the oral herpes, right? I'm lately infected, that and I can be shedding from my mouth. I should probably avoid kissing babies, especially newborn babies, because if I'm shedding the herpes virus in my saliva and I'm kissing the baby all over the face, right? That would be an opportunity for the virus maybe to get into the baby's eyes or nose or mouth, right? And then that could cause neonatal herpes, right? So again, folks, the skin, cutaneous is skin, ocular is the eye, and then it can invade the nervous system. It can be really serious. So we said we should probably start a tradition where when we want to kiss a baby, maybe kiss the baby on its toes, right? Something like that, right? But stay away. Don't kiss around the baby's face if you know that you have oral herpes infection. One more herpes virus you guys remember is the chickenpox shingles, the varicella zoster chickenpox shingles virus. So before the chickenpox vaccine, most of us would get infected as children, right? Sometimes really they could be really serious infections. Um, so there's a whole range of kind of like clinical signs and symptoms. But again, we'd usually get these fluid-filled vesicles, the so-called chickenpox, right? Maybe take a couple of weeks to get over if it wasn't a real serious infection, and then you'd get better. You felt great. But again, folks, the same old story. The varicella zoster virus, it invades the sensory neurons, and it's going to hang out in the ganglia along your spinal cord, your vertebral body, right? And then that means years later, those varicella zoster virus, they can reactivate, travel back down the axon of the sensory neuron, reinvade cells, epithelial cells, and you'll end up with this reactivation of the varicella zoster virus, which we call shingles, right? So here's the shingles, you guys. I haven't had it yet. My brother's had it, and he said it was horribly painful, right? Um, So one thing to remember is that in shingles, the reactivation, the fluid in those vesicles has infectious varicella zoster virus. So be really careful, you guys. If you have an outbreak of shingles, be careful. You don't want to be around, say, newborn babies. You don't want to be around anybody who's immunocompromised, right? Because you can be shedding infectious viruses. You're not as contagious as when you have chickenpox, because in chickenpox, even your respiratory secretions have the virus in them, right? So you're less contagious when you have shingles, but the virus in those vesicles is infectious. You have to be really careful. Now, they have developed a live attenuated vaccine, the chickenpox, and another one is for shingles. But this is a tough one, you guys, because live attenuated means the vaccine viruses, they can replicate, right? Attenuated means they've been weakened, right? So the idea with live attenuated virus vaccines, they inoculate you with the vaccine virus, and it's not supposed to cause harm, right? It's supposed to be weakened, so it's not supposed to cause harm, but you will be shedding the virus, the vaccine virus, folks. And again, this is really important because if you've been just vaccinated with a live attenuated, say, chickenpox vaccine or live attenuated shingles vaccine, you are shedding vaccine virus. So again, you don't want to get around little babies. You don't want to get around anybody who's immunocompromised, right? Because anybody who doesn't have a strong immune system, those weakened vaccine viruses can actually cause serious harm. So this next slide, you guys, I pirated it from a paper. So let's just take a look at people who should not receive live attenuated vaccines, okay? And I know this is hard to read, you guys, so um, the SID, I think, is severe immunodeficiency, and so there's different reasons why we might become severely um, deficient or immunocompromised. So folks, with HIV, you can have HIV and your immune system is still doing fine until the HIV destroys a significant number of your CD4 positive T helper lymphocytes. Those are like the most important cell of the immune system. So if I had HIV but I haven't progressed to AIDS, I could probably receive any vaccine, right? But if I have progressed to AIDS, meaning my T helper CD4 count is below 200 cells per microliter of blood, now officially I have AIDS, acquired immunodeficiency syndrome. So you should not give your patients that have AIDS any of the live attenuated vaccines. Their immune system can't control the replication of the vaccine viruses. Um, Blood and solid malignancy, so cancers here, you guys. So don't give live attenuated vaccines during chemotherapy. Patients that have bone marrow transplants, don't give the MMR, that's measles, mumps, rubella, or varicella zoster. These are live well, the varicella zoster for sure, you guys, is live and attenuated. Let me check into the MMR vaccine to see if that's a live attenuated. But for sure, you guys, you don't want to give the varicella zoster up to 2 years after the transplant, because your transplant recipients are going to be on immunosuppressive therapy. See, and don't and up to 3 months after immunosuppression therapy and chronic. Oh my gosh, I should know what this is, you guys. Okay, let me check. I should know this. GVHD. Grr. Okay, I need to check into that, you guys, so apologies. In solid organ transplants, okay, you just don't give them the live attenuated vaccines because they're probably going to be on maybe lifelong immunosuppressive therapy, right? So they don't reject the donated organ. In immunosuppressive therapy, for example, in autoimmune diseases, right? Some people have autoimmune diseases. So you wouldn't want to give the live attenuated viruses in your patients that are receiving high doses of immunosuppressive therapy, like high doses of corticosteroid therapy. Okay, and then asplenia. So sometimes maybe when we're kids, we fall out of the tree, right, and we rupture our spleen, so they take your spleen out. So you're going to be, for the rest of your life, somewhat immunocompromised. So again, folks, I think this is saying here that probably you'd want to avoid the live attenuated vaccines. And again, I'll have to double check this, you guys. Okay. So this is the last little bit, you guys. One concern with these latent viral infections is that some of them may increase our risk for certain types of cancers, right? And especially, you remember, you guys, with the Henrietta lacks HeLa cells, the cancer cells, the immortalized cells, you'll recall that the human papillomavirus strain 18 was DNA was found inserted into the chromosome of the cervical cancer cells, and it's believed that it was that HPV strain 18 that caused Henrietta lacks cervical cells to become cancerous, right? So when the virus genetic information gets integrated into our host cell chromosomes, it can cause all kinds of abnormalities in the cell surface of our cells and metabolic activities of our cells. And very importantly, you guys, it can really disrupt our cells' ability to control their growth and replication. So viruses can cause it, like if the virus invades a cell and causes it to start replicating over and over again, we would say that those viruses have induced or caused a tumor, so they're called tumor viruses. The cells they infect are called transformed, and the problem is, you guys, this can lead to cancer. So cells that cannot control their replication, they start invading, damaging surrounding tissues and organs, and then they might spread, metastasize and spread to other organs and tissues, and potentially can be fatal. And again, the concern is that some viruses we think, increase the risk of cancer. So they're called oncogenic viruses. And here's some examples. Folks, the first two I'd like you to remember. You don't have to remember the last one. But the first two, you guys, you probably already know that the hepatitis B virus, which is a DNA virus, and the hepatitis C virus, which is an RNA virus, um, they they like to replicate in our hepatocytes, our liver cells. And our liver is awesome, you guys. The liver cells, they can, like if a liver cell is damaged or dies, surrounding cells can replicate, right, trying to replace the damaged cells. But our liver cells can only replicate so many times. So if we've been chronically infected with hepatitis B or hepatitis C, we can end up with like a lot of fibrotic scar tissue that won't function as liver cells anymore. So we end up with liver cirrhosis, liver failure. And it also increased chronic infections with Hep B and Hep C also increases our risk for liver cancer, right? So this is bad news. The good news is, you guys, we have a vaccine available to help prevent infections with hepatitis B. So you could say this is a anti-cancer vaccine, right? If you don't get infected with hepatitis B, that's going to decrease your chance of developing liver cancer later in life. The bad news is, you guys, we haven't developed a vaccine against hepatitis C, and man, we need a hepatitis C vaccine as well. Here's our HPV human papillomavirus strain 16 and 18. In cervical cancer cells, it's incredible, you guys. Like 80 to 90% of cervical cancer cells have HPV DNA in the chromosome. So we know that these strains really increase our risk for cancers. So we'd call these strains oncogenic. And often you think of HPV as only causing cervical cancer in women, but the viruses, they really don't care, which epithelial cells are invaded. So certainly in women, they increase our risk for cervical cancer. That's why the pap smears are so important. But in men and women, you guys, it can increase the risk of anal rectal cancer, and it can increase the risk in men of cancer of the penis, penile cancer, and they're starting to see an increase in the number of throat cancers caused by these strains. And it's thought that this is because people think that oral sex is safer sex. And it's like, well, so if it was a man and a woman and they're having oral sex, you could say, okay, well, the woman will get pregnant, but you guys, these sexually transmitted pathogens, a mucous membrane is a mucous membrane to them. And with HPV, they don't care if it's the epithelium of the genital tract or the epithelium of your throat, right? So we're starting to see a big increase in throat cancers caused by these strains. So again, you guys, like educating your patients, it's so important. To me, you guys, the good thing is we have now at least two different vaccines. that can help protect against infection with HPV strain 16 and 18. The first one that came out was the Gardasil vaccine. Initially, it was just for girls and women, which to me was insane. It's like, because men, you guys, men can get these cancers from HPV strain 16 and 18, right? It made me crazy. It's like, why are you only using it to protect, girls and women? Our sons, our men need it. And so now, folks, men and women um, receive the Gardasil vaccine. And I know, folks, there's a lot of controversy over vaccines, but man, my thought was if, we could get our son and daughter vaccinated and hopefully prevent the chance of them developing any of these cancers. It's like, I was really gung ho about getting our kids vaccinated. Okay, and folks, this is another herpes virus family, not family. I'm sorry, you guys. This is another herpes virus, the Epstein-Barr virus, that is thought to increase the risk for cancer. This is the darling little virus that causes infectious mononucleosis, the so-called kissing disease. Now, it doesn't increase the risk for cancer in all people, so there must be different strains and maybe the genetics of the person that gets infected, but it can cause a cancer called Burkitt's lymphoma, and it can also increase the risk for nasopharyngeal cancer. And again, you guys, it's probably has to do with the strain of the virus and also host genetics, right? So again, folks, we want to just end on a good note. I mean, we can argue we do have some anti-cancer vaccines. The hepatitis B vaccine, by preventing infections with the hepatitis B virus, should reduce the risk of the prevalence of liver cancer worldwide. And likewise, you guys, our HPV strain 16 and 18 vaccines should reduce the prevalence of cervical cancer, anal rectal cancer, cancer of the penis, and cancer of the throat, right? So that is something to celebrate. Okay, folks, I know that was a long one, so let me just, I think that was it, right? Okay. So, folks, just so you know, this again, this is March of 2020. We're in the coronavirus pandemic. So, normally, before the coronavirus pandemic, we would have a second virus PowerPoint looking at specific RNA viruses, looking at influenza, looking at HIV, looking at poliovirus. But I think what I'll be doing this week is I'd like to work on a part two virus PowerPoint that explores how viral infections are diagnosed, look at some of the molecular biology that's involved, because I think it's really important we understand how is it they're diagnosing people infected with the coronavirus. We need to explore a really important concept of diagnostic tests called specificity and sensitivity. We want to talk about the limitations of the tests that they're using to try to detect the coronavirus. And we want to look at some of the weaknesses of some of the tests, right? And we also want to take a look at tests to detect antibodies against the coronavirus because, well, I'll leave that for the next PowerPoint, but I think there's some really, really important issues that we need to discuss with regard to diagnosis, detection, and antibodies against the coronavirus. So I'll stop here, you guys, and hopefully I'll have that next PowerPoint up maybe in like 2 days.